Hello children, in the second video of photosynthesis, I will be dealing with the first part or the first reaction called light reaction. So let us get started. We are all aware of the process of photosynthesis. Once again reiterating what you have learnt in lower classes, photosynthesis is a process by which plants prepare food using carbon dioxide and water in the presence of sunlight, and this sunlight is trapped by the chloroplast. And the end products of photosynthesis is glucose and oxygen. So this you are very familiar with. I am not balancing the equation. We are learning the substrate as well as the products on the other side of the equation. It is the production of glucose and oxygen utilizing carbon dioxide, water in the presence of solar energy, which is trapped by the chlorophyll present in the chloroplast. This is what is called photosynthesis. And we are very familiar with the part of the plant that does this. Photosynthesis is done by the leaf. Now, I am taking a cross section of the leaf, a section that is going through this part of the leaf. I am zooming that portion just to give you the idea of which cell is concerned with the process. The organ that is involved in photosynthesis is the leaf, and now we are going to see which cell is involved, and again we will zoom into the cell and see which organelle is involved. So the organ is the leaf. Very well you all know the leaf produces food by photosynthesis. Now in the leaf we are zooming to find out which cell. The cell is the mesophyll cell. So for that you have to have a clarity of the TS through the leaf. So I have taken the cross section or the transverse section through this part of the leaf, which is running through the vein. So you can see there are the upper epidermis, which I am drawing barrel shaped cells on top, and similarly down there is the lower epidermis, continuous layer of cells, lower epidermis, and in between the upper epidermis and lower epidermis there is the tissue called the mesophyll cells, which I will indicate in green color. And you all have learnt in 10th standard, mesophyll itself, there are two types of mesophyll, one which is arranged like columns, continuously arranged, they are called the palisade tissue, and just below that is a spongy tissue, they are circular and scattered. So the entire green portion is dealing with mesophyll, and the barrel shaped continuous layer of cells on the top and bottom are the epidermis. The upper one is called the upper epidermis and the lower one is called the lower epidermis. Anyway, we are going not into the detail of the anatomy. What I am trying to make you understand is which cell is involved in photosynthesis. It is the mesophyll cell. It is both the column shaped cells as well as the round scattered cells, which is a spongy cell. So we can see this mesophyll is sandwiched between the upper epidermis and lower epidermis, and it is in this mesophyll cell that photosynthesis is taking place. Again inside the cell, which organelle is participating in photosynthesis? You all definitely know it is chloroplast. So I am going to draw the location of chloroplasts. You have noticed that the leaf surface, on the leaf lamina, the upper surface, especially in the case of dicot leaf, the upper surface is very dark compared to the lower surface. The lower surface is not so dark green, it is light green. The lower lamina is light green, whereas the upper part of the lamina is very dark green. The reason is this, the distribution of chloroplasts. We can see dense distribution of chloroplasts in the upper part, that is in the palisade tissue. We can see the chloroplast is distributed densely, whereas the very same chloroplast, it is scarcely distributed in the cells, lower mesophyll cells called the spongy tissue. Anyway, whether it's a palisade or the spongy, these are coming under mesophyll cells, which are concerned with photosynthesis. They have chloroplast. I just told you, I gave you an extra information why the upper part is darker than the lower part. The reason is the upper part, which contains the mesophyll cells called palisade tissue, which is densely populated with chloroplast organelle, whereas the spongy tissue, which is in the lower coming below the palisade, has the chloroplast, which is scarcely distributed. Clear? So it is in the mesophyll. Anyway, it is within the epidermis, this full set of cells which are involved in photosynthesis. Now we are zooming into it. Which part? Definitely we know it's the chloroplast. So I pulled out the structure of the chloroplast. You all are well aware of the structure of the chloroplast. It is oval in outline, and there are two layers. The name is easy to remember: outer chloroplast layer, inner chloroplast layer, and inside there is a fluid filled compartment called the stroma. Then in the stroma are arranged the thylakoid. You can see each structure is the thylakoid, and the thylakoid is arranged in a stack, and the stack is called the granum. See, so you can see how many stacks you can see: 1, 2, 3, 4, 5 and 6. 6 stacks of granum, which is called the granae. And also another structure you can see, the granae are interconnected. See, they are interconnected with each other. The thylakoids of each granae are interconnected with some rod-like structures. These rod-like structures are called stroma lamella. Because they are located in the stroma, they are called stroma lamella. So these are the basic structures of the chloroplast. Now what I am trying to say, we are going to see how the process of photosynthesis is occurring in the chloroplast. There is division of labor in the process of photosynthesis. There are two very important phases in photosynthesis. The names of the two phases are: light reaction is the first phase, and the second phase is the dark reaction. So these two reactions are occurring inside the chloroplast, and this division, it's not that in one particular part of the chloroplast it's occurring. In one part light reaction takes place, and in the other part dark reaction takes place. Light reactions occur in the granae. So in the stacks the light reaction is occurring. So in all these stacks light reaction is occurring, whereas dark reaction occurs in the stroma. So there is a clear-cut division of labor. Light reaction occurs in the grana and dark reaction occurs in the stroma. Now further, with more differences between light and dark, I will be going to the next portion, but very importantly, what does this light reaction means? From the very term you can understand it is driven by light. It occurs in the presence of light. So the name is accurate. But dark reaction does not mean it occurs in the absence of light or it occurs in darkness. It's an inaccurate name. Then why is this name given? After light reaction, the next name that they gave is dark reaction. In this context, it's a misnomer, a very inaccurate name, but they put the name dark reaction because it is immediately following the light reaction to make a complete couplet. For example, we say boy, when it comes to boy we say girl. Father, what's the next? Mother. Day, night. Husband, wife. So it makes a complete couplet if we give a name which follows it. And because the dark reaction immediately follows the light reaction, it is given the name dark. And second reason why it is given dark reaction is the dark reaction depends upon all the products of the light reaction. So for that they gave the accurate name for light. It occurs in the presence of light or it is light driven, but dark reaction is a misnomer. It does not occur in darkness. Very clearly I am saying it does not occur in darkness or does not occur in the absence of light. It is given because it follows the light reaction, and not only that, the dark reaction depends on the products of light reaction. So that you should bear in mind. So now we are going to analyze some more differences between light reaction and dark reaction. Some parameters I have placed here where it occurs. I already mentioned light reaction occurs in the grana, whereas dark reaction occurs in the stroma. Next, what I mentioned is whether it's light driven or not. This point, light reaction is light driven, directly driven by light. But dark reaction is not at all driven by light. And you give a clause here, but depends on the products of the light reaction. And also it follows the light reaction immediately. And the third aspect we are going to see is the end products of light and dark reaction. Now, when you analyze this equation, you know the end product of photosynthesis definitely is, one is glucose and the other is oxygen. So remember, oxygen is the end product of light reaction, whereas glucose is the end product of dark reaction. So definitely the product should be present in the reaction. So oxygen is emitted during the light reaction and glucose is produced during the dark reaction. In addition to oxygen, what else is produced? Energy molecules like ATP and NADPH. So in light reaction, the end products not only include oxygen, it also includes the energy molecules ATP and NADPH. Whereas in dark reaction, we have glucose. And along with glucose, there is a substance that is also produced, but that is reused. It is regenerated and recycled and used. That is ribulose biphosphate. So once again, let's look into the difference between light and dark reaction, the two phases of photosynthesis. So photosynthesis has two phases. The two phases are light reaction and dark reaction. And let the first point be this: why it is called light reaction? Yes, it is directly driven by light. But dark reaction is a misnomer, is inaccurate, misleading, misguiding. It is not occurring in the absence of light or in darkness. It is not light driven, but it depends on the product of light reaction. So let that be your first point, which is going to justify the heading of the particular reaction. Then second point, where this light reaction occur, both the reaction occurs in the chloroplast, but light reaction occurs in the grana of the chloroplast, whereas dark reaction occurs in the stroma of the chloroplast. And lastly, the end products of light reaction. The end products of light reaction is oxygen as well as energy molecules like ATP and NADPH. And the products of dark reaction is glucose as well as another substance which is produced and forms the substrate. So we cannot say that is an end product because it is produced and that is utilized. Okay. So these are the points we can elicit between the difference between light reaction and dark reaction, and then only we are going to move into in detail about the light reaction. So you have to be very familiar with both the reactions or with both the phases, light reaction and dark reaction. And in today's video, I am going to elaborate on light reaction. Now before going into light reaction, I have given you an abbreviation of the energy molecule ATP, or let it be ADP. Its full form is: A stands for adenosine, D stands for di, and P stands for phosphate. So it is the full form for ADP. If it's ATP, just the change is instead of D it is T, which means tri. Okay, so the full form of ATP, which is the energy molecule, is A stands for adenosine, T stands for tri, and P stands for phosphate. If it's ADP, it is A stands for adenosine, D stands for di, and P stands for phosphate. At the same time, I told you about another energy molecule. The other energy molecule is NADPH. NADPH. N stands for nicotinamide. Nicotinamide. A stands for adenine. D stands for dinucleotide. P stands for phosphate and H stands for hydrogen. So, this is the second energy molecule we are going to learn in this chapter, and it is new to you. So, you should know the full form. N stands for nicotinamide, A stands for adenine, D stands for dinucleotide, P stands for phosphate and H stands for hydrogen. So, as I proceed, I will only use the abbreviation which is standardized. So, now we are going into the details of light reaction, as I told you. Before going into light reaction, we have to know the types of chlorophylls present in chloroplast. There are basically four different types of chlorophylls. They are chlorophyll A, chlorophyll B, xanthophylls and carotenoids. Chlorophyll A, chlorophyll B, xanthophylls and carotenoids. They differ in color. Chlorophyll A is bluish green, chlorophyll B is yellowish green. So, both are green pigments, but the shades are different. One is bluish green, the B is yellowish green, xanthophylls clear cut yellow in color and carotenoids orange color. So, we are going to see how these chlorophylls are arranged, and so we are going to explain, I am going to explain the light harvesting complex. Light harvesting complex. A light harvesting complex comprises of hundreds of pigment molecules. So, I am going to draw all the pigment molecules, hundreds of them, but at random I am showing a few of them scattered. Okay. Just assume it is a cluster of hundreds of pigment molecules, and these molecules are that of B's. B is yellowish green. So, I have taken here randomly, I have drawn yellow green here and there. Okay. We have chlorophyll B, then xanthophyll which is yellow in color, X here and there, and carotenoids which is orange in color, C. So, you can see the hundreds of pigments are located on proteins like a cluster, and this structure, what I have drawn now, this cluster is known as antenna. This entire structure is known as antennae. So, what is antennae? It is a group or cluster, association of hundreds of pigments, namely chlorophyll B, xanthophylls and carotenoids, not having chlorophyll A. Did I put chlorophyll A? No. Excluding chlorophyll A constitute the antenna. Now, I am going to, so you understood what is antenna. Now, there is a role for antenna. I will explain the role of antenna only after placing the chlorophyll A, and there is only one single chlorophyll A, and that single chlorophyll A is located at the center. And you know the color of chlorophyll A, it is bluish green. I am going for a blue color to make it stand out. So, this let it be chlorophyll A. Now, I added chlorophyll A which is located at the center, and chlorophyll A indicates the reaction center. Clear? Now, we have the entire light harvesting complex. So, what is a light harvesting complex? It comprises the antennae. I tell you what is antennae? All the pigments excluding chlorophyll A, and also the reaction center. What is reaction center? The single chlorophyll A which is located at the center. together constitute the light harvesting complex. Now, the speciality of A absorbs light of 680 nanometers or 700 nanometers of the light spectrum. So, once again chlorophyll A absorbs light either of 680 nanometers or 700 nanometers. If it absorbs light at 700 nanometers, this entire light harvesting complex will be called PS, photosystem 1. So, now this entire complex is called photosystem 1, PS1. Why? The specialty of photosystem 1 is the reaction center chlorophyll A absorbs light at 700 nanometers. Clear? But the very same structure, if the chlorophyll A absorbs light at 680 nanometers, I am rewriting over it, 680 nanometers, then it becomes another photosystem called photosystem 2. There the chlorophyll A absorbs light at 680 nanometers. So, we have two photosystems or two light harvesting complexes. They are PS1 and second one is PS2. The difference between PS1 and PS2 is only with respect to the chlorophyll A. In PS1, chlorophyll A absorbs light at 700 nanometers, whereas in PS2, the chlorophyll A absorbed light at 680 nanometers. So, that is the basic difference between the two light harvesting complexes or the two photosystems. So, I hope the two photosystems are well introduced to you. Anyway, basically a photosystem or a light harvesting complex comprises of two parts. I am highlighting, it comprises of the antenna and the reaction center. What is antenna? It is a group of hundreds of pigments excluding chlorophyll A. They are B and xanthophyls and carotenoids located on proteins, and along with the reaction center. Reaction centers represent only by one chlorophyll A, which is located at the center. Now, it depends. If chlorophyll A absorbs light at 700 nanometers, it is called photosystem 1. If chlorophyll A absorbs light at 680 nanometers, it is called photosystem 2. Now, why it is called photosystem 1 and photosystem 2? Here, don't misunderstand again, 1 because it is functioning first and 2 because it is functioning second during the light reaction. It is not based on the sequence of the function, it is based on the sequence of their discovery. So, the first to be discovered was photosystem 1 and later photosystem 2 was discovered. Clear? So, it is based on the sequence of their discovery. Now, as I told you, what is the role of the antenna? Very clearly, the role of chlorophyll A is mentioned, that is absorbs light at a particular wavelength. Now, the function of the other pigments in the light harvesting complex, what will be the function? These absorb light at various wavelengths. See, they absorb the sun's light at various wavelengths, different wavelengths, and they send it to the reaction center. All this is transferred to the reaction center. See, all the energy is transferred to the reaction center, and thereby they make the process of photosynthesis very efficient. So, the first function of the antenna. So, we are looking into the functions of antennae, that is all the hundreds of pigments surrounding the reaction center. The first function is they absorb light of various wavelength and transfers it to the reaction center. Reaction center is the chlorophyll A, and therefore, they make the process of photosynthesis very, very efficient. Next function is they protect. See, they all are surrounding the chlorophyll A. See, chlorophyll A is at the center. I am highlighting chlorophyll A. It is at the center. It is well protected. Chlorophyll A is very sensitive to light. If there was no, if these were not there, if these pigments were not there, what would happen? Due to the sun's light, this chlorophyll A would get destroyed, and that process of destruction of the chlorophyll by sun's light is known as photooxidation. Okay, if the pigment molecules were not surrounding chlorophyll A, what would have happened to chlorophyll A, the reaction center? It would have got destroyed by photooxidation. But now it is not being harmed by the sun's light because it is protected by the adjacent pigments, the pigments which are present around it, bees and xanthophylls and carotenoids, protect it from photooxidation. So this event, photooxidation, does not occur, it is well protected. So second, protects chlorophyll A from photooxidation. So with this, light harvesting complex is complete. Very briefly, I will give an overview of light harvesting complex. Light harvesting complex of photosystem. There are two photosystems, photosystem 1 and photosystem 2. The basic difference between photosystem 1 and photosystem 2 is with respect to the center placed chlorophyll, that is chlorophyll A, which serves as the reaction center. Basically, a light harvesting complex of photosystem comprise hundreds of pigments, bees and xanthophylls and carotenoids surrounding the center pigment, single pigment, the chief pigment chlorophyll A, which serves as a reaction center. So that full structure constitutes the light harvesting complex of photosystem 1. And as I told you, 1 was discovered first, and it was chlorophyll A which absorbs light at 700 nanometers, and photosystem 2 was discovered second, and it was the chlorophyll A of that light harvesting complex absorbs light at 680 nanometers. Now the chlorophyll surrounding the reaction center has a very major role to play. They protect the chlorophyll A from photooxidation, destruction of the chlorophyll A from sun's light, and second is they absorb light at various wavelengths and transfer that energy to chlorophyll A, which is the reaction center, and thus make the process of photosynthesis very efficient. Now we can move on to the light reaction. There are two types of light reaction. They are, first one is NCPP and the second type is CPP. The full form of NCP is non-cyclic photophosphorylation and the second type is cyclic photophosphorylation. So what is common to both the types is PP is common. So let me analyze, or let's look what is this PP. Now we saw energy molecule ATP is the energy molecule adenosine triphosphate. How it is formed? ADP, adenosine diphosphate plus phosphate combines to form ATP. This process is called phosphorylation. So what is phosphorylation? The addition of phosphate to adenosine diphosphate to form adenosine triphosphate is known as phosphorylation, and this occurs in the presence of sunlight. Therefore, it is called photophosphorylation. So definitely you know in NCPP and CPP what is happening? This event is occurring, that is ADP plus P in the presence of sunlight forms ATP. But we are going to see the difference. The difference definitely lies in the first abbreviation, NC, and the other one is C. One NC stands for non-cyclic and C stands for cyclic. To understand that, let me start off with the first type of light reaction called non-cyclic photophosphorylation. In non-cyclic photophosphorylation, both the photosystems are involved, photosystem 1 and photosystem 2. It is represented by an oval. First is photosystem 2. So don't misunderstand, as I told you, it was based on the sequence of their discovery, not on the basis of their function. So it is photosystem 2 that is first working. And what is photosystem 2? I will elaborate here. It is photosystem P680. 80 nanometers. We know by now what is photosystem 2. Now, due to the sun's light, photosystem 2 gets illuminated, and therefore an electron gets excited and jumps to an orbit further away from the nucleus. So, once again, when the sun's light illuminates the photosystem 2, an electron gets excited and emitted, and it immediately goes to an electron acceptor. What X is indicating is an electron acceptor. How did that electron go to that electron acceptor? When the photosystem 2 was illuminated, an electron got excited and got emitted and was accepted by an electron acceptor X. Now, from the electron acceptor, the electron travels through the electron transport chain. Electron transport chain is a chain of cytochromes. It passes through the electron transport chain downhill. Now, we will be using the two terms downhill and uphill. And remember, when we are using downhill and uphill, it is with respect to the redox potential scale. And the diagram itself will make you understand. Now it is moving downhill through the electron transport chain to the next photosystem, that is PS1. So, once again, there was an electron that was emitted from photosystem 1. Electron got emitted, and it got excited, emitted, and it is accepted by an electron acceptor X. Then this electron, from that electron acceptor X, it passes through the electron transport chain, and where will it go? It goes to the next photosystem, which is simultaneously illuminated by the sun's rays. See, the sun's rays is illuminating photosystem 2 at the same time, and therefore an electron is emitted from photosystem 2 also, which travels uphill to an electron acceptor, another electron acceptor X. So, an electron is lost from photosystem 1, but that electron that is lost is compensated by the electron coming from photosystem 2. See, the electron of photosystem 1 that is lost is compensated by the electron from photosystem 2. Now, this electron which is emitted from photosystem 1 goes to an electron acceptor, and from this electron acceptor it goes downhill to an energy molecule called NADP. When this electron reaches NADP, it gets reduced to form an energy molecule called NADPH plus H plus. Clear? So, we saw the electron, see the movement of electrons. The electron was emitted from photosystem 2 due to the illumination by the sun's rays. This electron gets emitted and passes to the electron acceptor X, the first electron acceptor, and by its location you can understand it's uphill. Then this electron passes downhill through an electron transport chain. And remember, what is an electron transport chain? It is a system of cytochromes. We will be learning later an electron transport chain. It consists of a number of cytochromes which are arranged in a series. And as the electrons are passing through the electron transport chain, it is not used. Remember, the electron is not used, it is just passed. And remember, very, very important process occurs here. That is phosphorylation. What is phosphorylation? ADP plus P gives ATP. So, the event called phosphorylation occurred here. When the electrons are passing through the electron transport chain, phosphorylation is occurring, and this electron is not used, it just passed. And then I hold on the electron here. Now, at the same time, what happens? The sun's rays illuminates photosystem 1, and an electron is emitted from photosystem 1, and that lost electron is replaced by the electron coming from photosystem 2. Now, the electron has reached the next electron acceptor uphill, and from this electron acceptor it goes to the energy molecule NADP, which gets reduced to form NADPH plus H. Now we can analyze the products. No, before that it's not complete. I told you how is this photosystem that has lost its electron has been compensated. Compensation has taken place here. The electron that was lost from here is replaced by the electron from photosystem 2. But this one's electron that is lost from photosystem 2, the electron lost from photosystem 2 should be replaced. How it is replaced? A very important event, that is photolysis. Water undergoes photolysis. It splits into oxygen atom and hydrogen and also electrons. And this electron that is lost, I told you, the electron that was lost due to elimination is now compensated by the photolysis of water. Now both the photosystem 2 and photosystem 1, the lost electron is replaced, or the lost electron is compensated. Now before I can give you an overview of this, we can just focus on the products of NCPP, that is non-cyclic photophosphorylation. I will highlight with a color which I have not used. Okay, first product is at this level, oxygen is one product. The next product is ATP, and the third product is NADPH. So I have highlighted the products. The products at the end of NCPP are oxygen atom. What else? ATP, energy molecule, and another energy molecule, NADPH. Then another thing, did you notice? Look at the path of the electron. Which path it is following? It started off with photosystem 2. It traveled, reached an electron acceptor, traveled down through the electron transport chain to the photosystem 1, then traveled uphill to another electron acceptor and went to and reached NADP. Okay, so it travels a non-cyclic path. So electron path is non-cyclic. Clear? So it's justified, it is a non-cyclic path. So now I am reiterating or recap of the process. First, photosystem 2 is illuminated by the sun's light. And when photosystem 2 is illuminated by the sun's light, an electron is excited. When it is excited, it is emitted and it is accepted by an electron acceptor. Clear? Then from this electron acceptor, now look at the location of the electron acceptor. It's gone above. Why? It is showing a greater redox potential. So remember, any product going up in the flow chart indicates it's of higher redox potential. And when the flow chart comes down, you use the word downhill. When the flow chart goes up, use the word uphill. Okay. So just remember, it goes uphill and it is accepted by an electron acceptor X. Now from this electron acceptor X, the electron travels downhill an electron transport chain to the photosystem 1. Simultaneously, what happens to photosystem 1? Photosystem 1 is also illuminated by the sun's light and an electron is lost. And this electron that is lost is replaced by the electron from photosystem 2. Now the electron that is lost from photosystem 1 goes to another second electron acceptor. Now look at the electron acceptor's location, it's also uphill because of a higher redox potential. And from this electron acceptor, it goes downhill to a molecule called NADP, which gets reduced to form NADPH. Now you remember, as the electron is passing through the electron transport chain, remember the electrons are not used but only passed from one cytochrome to another cytochrome, and meanwhile what is happening? The process of phosphorylation is taking place. ADP plus P gives ATP, and finally the end products we find is oxygen, ATP and NADPH. H, and the pathway of the electron is a non-cyclic pathway. Now moving on to the second type of light reaction. There are two types. It's not that the two types should occur in the same organism. It's either NCPP or CPP. CPP occurs mainly in bacteria. It involves only one photosystem, that is photosystem 1. That is the photosystem which absorbs light at P700. And you all are aware the bacteria is having a simple body design and can survive anywhere, and we have attributed that to the cell wall. But besides that cell wall, it is because of this. When it gets illuminated, it has a photosystem 1 which gets illuminated, which helps in the synthesis of ATP. That's why bacteria can live in very high light intensified conditions. So here the photosystem 1 is illuminated by the sun's light. And when the photosystem 1 gets illuminated, as I said, an electron gets excited, emitted, and it is accepted by an electron acceptor X. And then from this electron acceptor it goes back to photosystem 1 via the electron transport chain. So here also there is an electron transport chain operating where the electrons are passed. See, the electrons are passed through the electron transport chain. As it is passed, it's not used. What happens is ATP is generated, and you know the process how ATP is generated. ADP plus P gives ATP. Here, and finally this electron that is lost from photosystem 1 is replaced by the same electron. So, it is circulated within the system. There is no other systems involved. Photosystem 2 is not involved. So, this is very simple. Once again, only photosystem 1 is involved, and when as usual, when the sun's light illuminates the photosystem 1, an electron is emitted, gets excited, emitted, and is accepted by an electron acceptor, and from the electron acceptor it passes through the electron transport chain, and when the electron passes through the electron transport chain, what happens is phosphorylation occurs. We know by now what is phosphorylation. AP added to ADP, we get ATP, and it is returned back to the system. So, it starts from the system and returns to the system. So, see the pathway of the electron. The pathway of the electron is cyclic. So, these are the two types of two types of light reactions, NCPP and CPP. Both are light reactions but need not occur in the same organisms. NCPP is very common in plants and CPP is only in bacteria. But in plants also, besides NCPP, even CPP can operate. Now, we can give the difference. I have given already the parameters for NCPP and CPP when it occurs. Remember, when light of wavelength 680 nanometers is present, whereas CPP occurs when light of wavelength more than 680 nanometers is present. That means when there is high light intensity, CPP occurs. Whereas when the intensity is 680 nanometers, which event occurs? NCPP. That is why all the plants show NCPP, but bacteria can show CPP when at high light intensity like 700 nanometers, it will be operating the CPP. Next, where does it occur? Now, where? Here I told you light reaction occurs where? In the grana, and because we are talking about plants, photosynthesis occurring in plants, that is why we gave in the beginning itself light reaction occurs in the grana as a generalized statement. But when it comes to bacteria, it is not occurring in the grana, it is occurring in the lamella of the stroma. Grana. In NCPP it is occurring in grana, here it is occurring in the stroma lamella. Because it is bacteria, when we gave the generalized version of light reaction, I told light reaction because we are dealing with photosynthesis. So, light reaction photosynthesis means this is for plants. So, I give it as grana. Next, why it is occurring here? here in grana and there stromal lamella. Why couldn't it operate there also in grana? The reason is that the granule membrane, the membrane consists of what and all is absent here, here it consists. So, the membrane consists of all the photosystem, photosystem 1, photosystem 2 and an enzyme which helps in the reduction of NADPH. That is, the enzyme is NADP reductase enzyme. So, you can easily fill in the side. It lacks, what and all was missing in the diagram? It lacks photosystem 2 and that enzyme, reductase enzyme, NADPH reductase enzyme. So, the membrane, remember the membrane lacks. So, here the lamellar membrane lacks. Next, electron pathway. That is very simple. The electron path you saw, it took a non-cyclic path. In the first case, in NCP it took a non-cyclic path, whereas in CPP it took a cyclic path. And lastly, what are the end products? This I will highlight it with another color. The end products are oxygen, energy molecule ATP and NADPH. Here only ATP is the end product. So, you got the difference between NCPP and CPP.